Our next speaker is Carolina Chavarro, who is not able to present, and so presenting on her behalf will be Soraya Bertioli. And the talk is entitled "A Tree of Relationships of Section Arachis of Unprecedented Scope and Detail." So Soraya, if you could share your screen.
Okay, it's not sharing, is it? Okay, so I'm sharing the wrong screen. Swap displays.
Yep.
Oh, yeah.
That should work. There you go. Perfect.
Excellent.
All right. Thank you, Soraya.
Okay, so let's go to the first one. So yes, well, thank you very much for the chance to be here. I'm also going to give two talks. One of them is on behalf of Carolina Chavarro. She was a postdoc for a few years now. And fortunately for her, unfortunately for us, she went on to Bayer. So she went on to bigger things, and we're very happy to say goodbye, I suppose. So this work, she's done the heavy lifting. What I'm going to show here very quickly is what we've been doing for the last 3 years. And I presented part of it last year. So what we're doing is we are, there's more and more talk and more importance and emphasis is being given to wild species. And we know why. We're seeing more and more wild species being used for broadening the narrow genetic basis of peanut and helping the future of it. So I think the first wild-derived cultivar, the first example was corn that had resistance to root knot nematode, and it was actually the parent of several modern cultivars. And it is derived from crosses made by Dr. Charles Simpson, who got an award, very well-deserved award last week for a lifetime dedicated to peanuts, which is very nice. So several examples are present. So Bailey, for instance, it's very well-known cultivar in North Carolina, much grown and really with strong resistances, especially to early leaf spot. That's derived from a different cross, the hexaploid root. This one is a bit of a segue from what Dave was talking about. Last year, the cultivar Sempre Verde, which means always green, was released in Brazil, and it has segments of Arachis cardenasiae. It also comes from a roundabout way through India and to Brazil from the cardenasiae hexaploid root. And it is the first cultivar in Brazil recommended for cultivation without control of leaf diseases. It yields the same as the best local cultivars. Slightly different, Ipaensis and duranensis, an example in Senegal. Some of the wild-derived lines or cultivars, 6 cultivars were released. They have slightly higher yield, and they have good yield stability under drought. So this is, is huge for Senegal, and also slightly better resistance to early leaf spot. So we are tapping furiously into these new sources of resistance. So we've been involved in over 20 allotetraploids now, 5 of which were released last year between us and Peggy Ozaya's group. There is more to come. So why do you want to know about wild species? I mean, we know it comes from South America. There's about 92 described species. They're very diverse. They come from different environments, different climates. They have different morphological characters, and they have a lot of potential to give to our current peanut culture. So no one knew very much about the wild species until 56 years ago. The collection trips started in 1959 in South America. Some of the early people making the collections were Antonio Krapovickas, who some of us had the pleasure to meet, and Walton Gregory from North Carolina. So this was consolidated, the knowledge was consolidated into the, we call it monograph, which is like the Bible of the wild species. So he has, I think, 68 species that were known at the time. And it was translated by Williams and Simpson. And he divided the Arachis genus into 9 different sections. And these shows the relationships. Each of these lines basically shows which section is kind of crossable with a different section. So you can see the Arachis section is possible to cross with Erectoides and Rhizomatosi, but obviously they don't give good fertile hybrids. So probably the most comprehensive study so far was done by Morezzo, or we involved in it in 2013. And if you look here, there were just a very few accessions done, very few markers. And at the time, we managed to separate the B genome species from the A genome species. And you see these D, F, and K were like intermediate. And the Arachis genome, Arachis hypogaea A, grouping with the duranensis, so confirming the origin, and Arachis hypogaea B, grouping with Ipaentus, indistinguishable in this case. And later on with the sequence, we saw that we confirmed that with the genome sequence. So the objectives of the work we're doing still here is characterize, classify, and confirm identification of wild species, which is very difficult, very, based mostly on morphological traits and crossability. And very few people can do that in the world. We're helping identify new species and confirm species assignments. We check for the label mutations, which means it's very easy to swap seeds. I mean, everyone does that. It's, especially because of geocarpic, sometimes we pull something up and it's not quite the same. Anyway, these seed mixes are easy to happen. So we have identified and purged lots of seed lots. And another thing we want to do is to make a specific set of markers for different species. So we're going to have some haplotype type markers. And also we want to have guides for the choice of production of new allotype diploids. And I'll explain in a minute why. So what we did, we gathered accessions or DNA mostly, or leads from the main gene banks, all of them except Picrosat, that we had more difficulty with exchanging material. But we got from the 2 origins, EMBRAPA and IBONE. So when the collections were done, originally, part of the samples stayed in IBONE, some stayed in EMBRAPA, some were sent to Texas, and some were sent to USDA, and some to Picrosat. So what we know is that these, the less derived, they're probably the most likely to have had any seed mix because they haven't been transported. So we combine, all this knowledge of these original banks, these curators, with the new technology of the Axiom Thermo Fisher, we call it AFICHIP. It was originally by Clevenger and Pandy, and the version 2 was from Korani's work. So very, very summarized. I mean, summarizing the work of 3 years is this. Again, we find that B genome clusters with B genome species, the A genome species cluster with A genome species. We have the intermediate genomes clustering together here. So we find that it's quite similar to what we got some years ago. And if you observe this tree, you see that it's not rooted. So we we didn't put any common ancestor or anything. So we decided to put some outgroups there. And what we found, instead of rooting outside the tree, these were accessions from these 3 different sections. So we expected it to be completely as an outgroup, and actually it blended within the other species. So we have still a lot of work to do, but it gives us the idea that this is actually a polyphyletic group. And the next thing we're going to do is to put some Styloxanthes, which is the most close related genus. So we see if we can root the tree and identify common origins. And we're also going to expand this work. We're expanding this work with other species of the different Arachis sections. We want to show today, which is again part of the next talk, the case of Arachis stenosperma, which is where this big arrow is. Why we're interested in Arachis stenosperma, we're going to see a bit more later. It provides a lot of resistance, and we've been working with it for a while now. So Arachis stenosperma was collected basically in very 2 different distinct regions in Brazil. You see different clades here. You have a very distinct clade here, and 1 more original clade and 2 derived clades. And this actually compares very well, correlates very well with the geographic region. So this original clade, it's this one here on the top of Mato Grosso, where probably were the first accessions arose. And we have another clade that is more to the south. And the most derived clade is here in the coastal region of Brazil. And there are many evidences that, you have the same species here and here. So they were transported by native Amerindians. And so this corroborates this hypothesis of the dispersal of these species by native peoples. So basically, based on the objectives, we've come a long way. We've characterized most of the wild species, certainly all of the one of the Arachis section. And we then helped confirm, identify new species that are being published now. So these Arachis inflata, look how sweet it is. Guillermo Seijo from Argentina has just published work in collaboration with Dr. Simpson and other authors. We purged lots of accessions that had wrong labels. We are, as fast as we can, developing sets of markers that are species specific, and that's going to help all of us who work with wild species. So Samuele Lamon is going to take on this work and is guiding us make choices for the production of new allotetraploids. And the reason being this: if you look at the tree again, you have lots of these stars here. So the red stars indicate where are the accessions where we already have allotetraploids produced. So these regions here, we know that they're covered. We have a lot of the allelic diversity represented in a tetraploid form that can be used by breeders. The blue ones are allotetraploids in progress, but in green is the ones that the clades that are not represented. So why do we want that? So as Tim Widmer said earlier, we need to be proactive rather than reactive. So let's make this diversity available so that we will have something to go on to when we have new epidemics. So to finish this part of the presentation, I'd like to, to thank the organization. It's always lovely to present at APRES, and thank Carolina Chavarro for all her work and the collaborations who have been amazing, contributing their knowledge and experience for us to move on with this. And I'd like to say that we're hiring. So if anyone is interested in working with us, please send us an email that we would love to hear from you.